Okay, so the example we went through, that's known as a test cross, where you cross an individual with a known phenotype and unknown genotype by a homozygous recessive individual. So in that one generation of cross in the offspring, you can tell what the genotype was of the parent based on whether the offspring are all with the dominant phenotype or half with the dominant, half with the recessive phenotype. So we'll see some examples of test cross as we go along, and we explored the outcomes for test cross involving one gene. You also could do this with multiple genes, and we'll do that later in the semester. So you can extend this predicting of outcomes into other possibilities, and in a kind of a standard genetic investigation, you typically want to know a lot about the genotypes of the individuals you're crossing. And you can make predictions of their offspring, very accurate predictions, if you know the genotypes of the individuals that you started off in the cross. So let's say we take a dwarf individual here and a heterozygous tall individual with their genotypes as shown. We could take their diploid genotypes and figure out what their gametes, the haploid gametes, would look like. So whether that individual is homozygous or heterozygous is going to influence what kinds of gametes it can make, how many unique kinds of gametes it could make. So if we start with the dwarf individual, that dwarf individual is homozygous recessive for the T gene, has only the recessive little t allele, and therefore every gamete that it makes is going to have that little t allele. So we actually know a lot about the kinds of gametes this can make, and the predictions are maybe not so exciting because they're 100% little t. If we have somebody who's heterozygous, then each of those alleles of the two kinds that we have, they each have an equal chance, an equal random chance of ending up in a gamete. So if you are big T and little t in a diploid, when you go on to make a haploid gamete, then that gets either the big T or the little t. So this is one way to think of it, is you sort of take a random pick out of the possibilities. By the rules of meiosis that we'll see later in the semester, the gametes produced that have a big T are equal in number to the gametes that are produced with the little t. And so however many gametes you make, roughly half of them should be big T and half of them should be little t. So predicting the odds of ending up in a gamete, we can do that with these guys over here, which will be 100% little t, and over here would be 1/2 or 50% big T and 1/2 or 50% little t, right? Not too hard to figure that one out. So then the genetic tests and predictions move forward by combining those gametes into offspring, potential offspring. So we can predict the offspring if we know what kinds of gametes can be made. So on the left parent, this dwarf parent can produce only little t gametes. And so again, that's 100%. The tall parent can produce big T gametes or little t gametes. And so this is one of the possibilities. Little t from the dwarf parent combines with little t from the tall parent. So this was bound to happen. We knew this was going to happen. There's really no other way for this to happen from the dwarf parent. They have to give a little t. And then that outcome also depends on having a little t from the tall parent, and that would be half of the possible gametes, and so that's half of the possible zygotes that are made, right? So there's basically a half chance of getting little t, because the only other possibility is big T from the tall parent, and that would be big T, little t for that outcome. And so we can predict, I think I pretty well solved that one for you. So half chance of getting little t, therefore half chance of getting big T, little t. So the same thing here to answer this question, left parent, all little t. And I'll switch to fractions here. Half of the gametes here are big T. So to get a big T, little t offspring, that would be half of the possible outcomes from this cross. All right, let's complicate it a little bit more. We can look, instead of genotypes, and look at phenotypes instead. So for the big T or little t alleles, we have our possible genotypes. So I'm just going to write out the genotypes that can give us tall or dwarf individuals. Sorry, I do not have an eraser on here. Little t, little t. So these two are would generate tall, and this one would generate a dwarf offspring. And one of those is not actually possible from this cross. So it's not possible to generate the individual that has a big T from each parent because the dwarf parent can't give a big T. So we actually have two kinds of gametes, or sorry, two kinds of genotypes that can be made, and those correspond to two different kinds of phenotypes that could be made. So this is a 50% or 1/2 chance that the offspring would be tall. Don't worry, it gets more complicated. All right, let's say we cross these individuals in a Punnett square. You can take all the possible, all the possible gametes that could be made from each parent and put those in combination. So we did the little example with the Punnett square on the test cross. Now we'll do an example here with some other genotypes, known genotypes. And so you can combine them in basically one parent shows up on the top and one parent shows up on the side. It's usually up to your choice. Obviously, you want to keep track of which one you're doing. This is, I think, tempting. If you have a single gene for diploid individuals, it's tempting to write this 2x2 Punnett squared. And if you do that without thinking about it, I want to try to break you of that this semester. So when you write little t and little t, you're really writing the odds that little t is going to happen is equal to the chance that little t is going to happen. Well, you've just repeated yourself. So you're going to repeat the effort and the writing, and it's really unnecessary. And if you get savvy about this, you can save yourself some time and simplify this down, because you know that this individual has only one kind of gamete it can make. So this is a word I'm going to use a lot: unique kinds of gametes. So there's only one unique kind of gamete that the dwarf parent can make. The tall parent is heterozygous, can give us two different kinds of gametes. And so we can solve that just like we've done before. Half chance, so half of the possible squares are big T, little t, and half of the possible squares are little t, little t. We can put in genotypes and phenotypes. And depending on what you're interested in for the outcome, depending on what you get asked on an assignment or exam, you could answer that question any number of ways. All right, so that's pretty straightforward. And what we've done up to this point deals with a single gene at a time. We're going to combine two genes in a second. Before I want to go too much farther, I want to talk about this term. This is a monohybrid cross. And this is kind of a standard genetic cross. So if you're doing a monohybrid cross, that involves crossing individuals from different genetic backgrounds and basically doing this genetic cross to try and figure out how the trait is related to the genes that are involved. So we sort of know a lot about these because of how they've been worked out, but you could do this with an unknown new phenotype and try to figure out the genes responsible. So this is a nice early process you can do to figure out the genetic control of a phenotype. So I know we've been working with tall and dwarf pea plants. Let's forget what we know about genetic control of tall and dwarf pea plants for a second and say we just knew that there were some tall pea plants from a group of pea plants that were always tall and some dwarf pea plants from a group of pea plants that were always dwarf. And we crossed them together and found out that their offspring across the board were 100% tall. So this was one of Mendel's, I guess, important discoveries, this idea of complete dominance, that you can have two different phenotypes, cross them together and end up with a phenotype that is exactly like one of the parents and really nothing like the other parent. And of course, we know how this works now, and I'll spell that out in a little bit. Well, let's do it now. So we've covered the genes for these. We knew that we had true breeding parents to start with, so that would be heterozygous dominant crossed with heterozygous homozygous dominant crossed with homozygous recessive. Sorry, there's a lot to keep track of when I'm writing on a screen here. All right, homozygous dominant crossed with homozygous recessive got us heterozygous, big T from the tall parent, little t from the dwarf parent. We don't have to do a Punnett square, but we could. Punnett square would involve one kind of gamete from the tall parent, one kind of gamete from the dwarf parent, and just reinforce the idea that there's only one way that those can combine to get us a diploid offspring genotype, which is tall and heterozygous. All right, so moving on, if you cross two of these F1 tall pea plants, so this is part of the monohybrid cross, is this terminology of the parent generation, the P parental generation, and the F1 generation, the first filial generation. So that refers to kids. So the F1 generation, and then to get the F2 generation, you cross your F1 by another F1. So siblings or individuals from the same kind of cross. And crossing them together, you then, if you were Gregor Mendel, discover that the phenotype from your first parents, one of those phenotypes that was lost in the F1, this dwarf phenotype, shows up again, just as it was in the parent. So something is sort of lost and then regained exactly as it was before. So a very important thing to learn about and discover, what was it that was being passed on? Well, of course, we now know that this dwarf individual recovers the little t homozygous recessive genotype by getting a little t from each of the parents in the F1 generation. So that's possible, but it is a minority of the outcome. So these other 3 out of 4 individuals are tall pea plants, and 1/4 of them are dwarf plants, with a pretty reliable 1/4, 3/4 ratio. All right. So this expected F2 offspring ratios, this is something that we now understand very well. If you do a Punnett square of heterozygous tall by heterozygous tall, now we get a little more exciting Punnett square. So here we have dominant, dominant, dominant from the left parent, recessive from the top parent, dominant from the top parent, recessive from the left parent, and recessive from each parent. And I just want to reinforce here that I'm writing the big T first. So I've got these diploid genotypes. They got their big T from different parents, but they end up having the same genotype, and that's effectively the same regardless of how their genes have combined. So rather than writing, say, little t / big T, which is ripe for confusion, this is a better way to write it. So these are the outcomes. These are the same, but it's important to keep track of both of them because this says that 50% or 2 out of 4 of the possible outcomes are going to be heterozygous. 1 out of 4 is going to be homozygous dominant. 1 out of 4 is going to be homozygous recessive. And maybe more importantly, if we're looking at phenotype, 3/4 tall and 1/4 dwarf. And then this also sometimes gets written as a 3-to-1 ratio. And I'm not super fond of writing it that way because I think writing 3-to-1 gets you to forget the denominator here. So 3-to-1 really means 3/4 of one kind and 1/4 of another kind. It means 3 times as many tall plants as there would be dwarf plants. Or you could think of this as percentages or decimals, 75%, 25%. We're going to get comfortable with our numbers as we do this. So 75%, 25%, 0.75 to 0.25, all different ways of writing the exact same thing. Okay, so this is the result of a monohybrid cross. And for genes that behave as Mendelian genes, this is the outcome that you can expect pretty reliably from one of these crosses. So completely dominant genes. All right, let's complicate it up a little bit and do a dihybrid cross. So a cross between 2 individuals that initially are different in 2 kinds of phenotypes. So here we have color, green versus yellow, and also texture or roundness, round versus wrinkle. And so those, as Gregor Mendel knew these are controlled by independent genes. There's a gene for color and there's a gene for round versus wrinkled. And you can combine those together and observe that the rules for transmitting the color gene and alleles, those are the same rules and the same processes that are transmitting the roundness or wrinkleness phenotype. So same thing as before, crossing these individuals discovered that the F1 offspring are uniformly the same phenotype. And in this F1 generation, this tells us who is dominant. So of the color, green versus yellow, yellow is dominant. So you could write your alleles in as big Y for yellow. And for me, I like if I have letters in my handwriting that are similar in uppercase and lowercase, give them a little curly Q or something that makes them distinct. So I can tell this is a lowercase y and this is an uppercase y. So I chose the letter for this. Usually it's kind of common, I guess, to choose the color for the dominant phenotype. So big Y for yellow and little y for green. Now, round and wrinkled, which one is dominant? Turns out here that being round is dominant, right? Because we see that showing up in the offspring. All right. And then as we cross them, the F1 individuals are going to get a mix of dominant alleles and recessive alleles. The big Y makes them yellow, the big R makes them round, but they are heterozygous because they had different kinds of parents here. So everything still checks out. You could look at the genes independently. You can look at color and see that yellow by green gets you yellow. You could look at roundness and say round by wrinkled gets you round. Those things are still true even though we're looking at both genes at the same time. So that's kind of important. We're going to see that this independence is kind of important. All right, so what does our F2 generation look like? Well, it gets a little more complicated. And you can do a bigger Punnett square here. So this is our, sorry, I put Y as first. They've got their R's first, but same thing, heterozygous for the roundness gene and the color gene. And you could do a gamete square and figure out what your gametes would look like. One allele for the R gene, one allele for the Y gene, big R with big Y, big R with little y, little r with big Y, little r with little y. And then those in a Punnett square would be the same for each parent, right? So across the top, you have the 4 possible kinds of gametes for one parent, and across the side, you have the 4 possible gametes for the same, really genetically the same parent there. So what happens? Well, same thing happens as before. We're going to combine our R's together into diploid R genotypes and our Y's together into diploid Y genotypes. And then those translate into phenotypes. So who are the round P's? Well, the round P's are the ones that got at least one of the big R allele. So homozygous dominant, big R, or heterozygous big R, little r. And that ends up being 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. So 12 out of a total of 16 that are round. And that fraction is the same as 3/4. So see that coming up where the ratios are the same for these genes, but when we combine them in combination, we look at round and color, we're going to get a combination of a few more different kinds of ratios here. All right, so we can look at these independent, and we can look at them using the genes. So let's say we do our cross, and we were crossing plants that make, let's see, our heterozygous plants were, we had big Y, little y, big Y, little y. Those were the F1 parents, cross them together. And looking just at the color gene, we predict that green is this recessive phenotype, 1/4 chance of getting a green P. And sure enough, that's true. So 1, 2, 3, 4 of the offspring produce 4 out of 16, I should say. So a ratio. 4/16 or 1/4 of these are green if we're looking only at color, green versus yellow. If we look at roundness, then we can look at same thing, same kind of Punnett square for the round and wrinkled. Round, wrinkled, round, wrinkled. All right, so round here, we predict 3/4. And let's see how many of these are round. We already counted that up. That was 12 out of 16 round, and that's the same as 3/4. So these are still the same ratios we would predict if we were looking at one gene at a time. Let's say you're asking the question, what if you got a plant that had green seeds and round seeds, right? So this is where the Punnett square comes in handy, saves you maybe thinking about these complex multi-gene possibilities for the phenotype. You can just draw out your Punnett square, figure out how the genotypes contribute to phenotypes, and then just figure that out. This one's nicely illustrated for you. So plants with green seeds and round seeds, that was 3 out of 16. So 3/16 green and round. And we're going to see that is related to the combination of green and round. Another way to look at that, so we'll have a chance to see that play out mathematically probably next time around. Another way to look at that, maybe a little more graphically, is you could look at the possibilities as independent but sort of sequential. So I've drawn these as boxes that are more or less sized according to the proportion of the outcomes that go along with that. So if we look at our color gene first, there was 3/4 chance that the plant would produce yellow seeds, 1/4 chance it would produce green seeds. And then out of those outcomes, we can subdivide them again. So 3/4 of the 1/4 are round. So that would be green and round. And you can multiply those together. 1/4 times 3/4 tells us 3/16. So we multiply the numerator, 1 times 3 gets us 3. Multiply the denominator, 4 times 4 gets us 16. 3/16 round and green. Same number comes from getting 1/4 of 3/4. So 3/4 times 1/4 is also 3/16. Yellow and round, 3/4 times 3/4 is 9/16. And green and wrinkled, 1/4 of 1/4 is 1/16. And so the commonly written ratio that you expect from a dihybrid cross is 9, 3, 3, 1, or 9/16, 3/16, 3/16, and 1/16. So you could figure that out by adding 9, 3, 3, and 1. That gets you 16, but this is maybe a cleaner way of writing that. These don't make nice clean decimals like we did before, but you could calculate that into a decimal as well. So this is the expected frequency to get the dominant phenotype for roundness and the dominant phenotype for yellowness. As you might expect, these were the most common outcomes, and they are the most common. Just over half of our outcomes are the combination of round and yellow. And then getting one dominant and one recessive outcome, those are equally likely 3/16, and then getting both recessive outcomes, 1/16. Okay, so a lot of people are comfortable with the Punnett squares. I think you should probably get comfortable using a Punnett square. Some people like these branch diagrams. They get you to the same answer. And so I want to be a little flexible in this class as to how you get to your answer. Some people are not super comfortable with the numbers and the multiplying and the fractions, and that's fine. That would probably get you through this class pretty well. And they might prefer to do the Punnett squares. Other people are comfortable with the numbers and they just want to dive right in and start multiplying. Up to you. All right, so that's the end of Mendelian genetics. Next up, we're going to combine, get a little more complicated in our genetic predictions.